Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall discuss regulation of enzyme amount. In the previous videos, we discussed how we can regulate metabolism by changing the catalytic activity of existing enzymes. In today's video, we shall focus how we can regulate metabolism by changing the amount of enzymes within a cell. So we can regulate the amount or the concentration of enzymes within the cell either by regulating the synthesis of new enzymes, or we can regulate how fast the existing enzymes are being degraded by the cell. So we could regulate synthesis, or we could regulate degradation of enzymes, which are proteins. So first, let's look at how we regulate synthesis of new enzymes. Since most enzymes that are involved in metabolism are proteins, we discuss how proteins are synthesized and transported in video 3.4. So this would be a good video to go back and review before you continue with this video here. Recall that making a protein is a relatively long process. It involves many steps that includes chromatin remodeling, getting the information from DNA and transferring it onto RNA, what we call transcription, and then the RNA has to undergo RNA processing, and then the matured RNA is sent out into the cytoplasm. And in the cytoplasm, on the ribosomes, the messenger RNA is translated, the protein is made, the protein folds, and then some proteins undergo post-translational modification. Once the protein is made and modified, it is transported to various parts of the cell, and that's called protein transport. So the amount of a given enzyme in a cell really depends on how fast these steps are occurring. The faster these steps occur, the more the concentration of the enzyme. The slower these steps occur, the less the concentration of the enzyme. There are many different proteins that are involved in these steps that lead to the synthesis of an enzyme. This means that in order to control how much enzyme is present in the cell, any one of these proteins that are involved in these steps can be regulated. Let's look at some examples of these proteins. Transcription factors are proteins that bind promoters as well as enhancer sites on the DNA, and that initiates the process of transcription. These transcription factors, like all proteins, are synthesized in the cytoplasm, and after they are synthesized, they have to be transported into the nucleus in order to function. So one way to control the amount of enzyme present is controlling the transport of transcription factors from the cytoplasm into the nucleus where they act. DNA bending proteins are proteins that bring enhancers close to promoters. That's another set of proteins that can be regulated. Different tissues have different promoters, alternative promoters, and that's yet another way to control how much enzyme is present in a certain tissue. After transcription factor binding, the promoter is then prepared for transcription, and for this, helper proteins are required. Examples of helper proteins include chromatin remodelers, co-activators, as well as general or basal transcription factors, and these proteins help remove the histone obstruction, help recruit RNA polymerase, as well as stabilize the protein transcription machinery. So all of these helper proteins could be regulated, and that would affect how much enzyme is present in the cell. So the next step, the RNA polymerase will now come and bind to the DNA. So that's Pol 1 for ribosomal RNA, 2 for messenger RNA, and 3 for transfer RNA. And then the mediator proteins will come and bind and help strengthen the binding of the RNA polymerase, as well as increase the polymerase activity. So it's not surprising that the regulation of mediator proteins will also affect the concentration of an enzyme in the cell. Other proteins such as transcription elongation factors, which are also required for transcription, can be regulated and thereby affect the concentration of the enzyme in the cell. Once the RNA is made, then the RNA undergoes RNA processing. Examples that we discussed were splicing, addition of cap, the addition of a tail, and these steps require processing proteins. So obviously the cell could control the activity of its processing proteins and thereby affect the concentration of the enzyme in the cell. After the RNA is processed, the matured RNA is then transported out of the nucleus by transport proteins. And once again, we can control the activity of these transport proteins and thereby affect the concentration of the enzyme in the cell. Once the RNA are in the cytoplasm, then the messenger RNA is translated into protein, and that some proteins then will become modified, what we call post-translational modification. Eventually, these proteins will be transported to various parts of the cell. So all of these processes, these steps here require a variety of different proteins. For example, protein synthesis requires the enzyme amino acid tRNA synthetase that's involved in charging tRNAs. Protein synthesis requires initiation factors, elongation factors, release factors, and so on. So any one of these proteins that are involved in these steps can be regulated in order to control the concentration of an enzyme in the cell. One of the biomolecules that seems to play a very important role in controlling the amount of an enzyme in a cell, what we say regulating gene expression, is these RNA molecules that are called long non-coding RNAs or LNC RNAs. These are long transcripts greater than 200 nucleotides. They do not code for proteins, but they're involved in numerous steps of gene expression, including histone modifications, chromosome structure, transcription factor recruitment, RNA processing, mRNA stability, translation, post-translational modifications, and so on. So there's a lot of interest in understanding these molecules because they actually control how fast the gene is expressed, which means how much of an enzyme will be found in a certain cell. So that's a quick overview of changing the amount of an enzyme by regulating synthesis of the enzyme molecule. Now, the concentration of a given enzyme also depends on the degradation of the enzyme. So not only does it depend on synthesis, but it also depends how fast the enzyme is degraded. So we discussed protein degradation in video 5.28, protein breakdown. We discussed the ubiquitin proteasome system, or the UPS, as well as the lysosomal autophagic pathway. So how fast these pathways occur will decide how quickly the protein is broken down and therefore affect the concentration of the enzyme. So we're not going to discuss this here. We recommend that you look at this video 5.28 if you are interested in the protein breakdown aspects. So thank you once again for watching and supporting this channel. Until the next video, have a wonderful day, and hopefully we shall see you soon.